Great. So hi, everybody. Welcome to the Single Cell Spatial Analysis Program seminar series. I'm Justin Colosino. I'm faculty in Environmental Health Sciences and Nutritional Sciences at the School of Public Health. And I'm one of the co-leaders of this University of Michigan Biosciences Initiative support program. And so if you're not a member of our program and are interested in joining or learning more, we'll put a link to our website in the chat. And today we're in for a treat. So we have some colleagues from Isoplexis, Brad Annen and Noah Vanderhyde, who are here to tell us more about their single cell proteomics methods and provide us with a hands-on demonstration of the capabilities of their software. And then make sure to stay tuned until the end, as they'll be announcing an exciting pilot grant program to generate some data using this platform. And so if you have any questions during the talk, please feel free to add them to the chat, and then we'll have Brad and Noah answer them. And with that, I will turn it over to Brad. Thank you so much.
Awesome. Thank you, Justin. First of all, I want to thank everyone for helping set this up. Dr. Keller, and then Vanessa, it's been a great pleasure to work with you, get this set up, and then the exciting grant program that we'll touch on a little bit later. So first off, with some thank yous there, and then also want to thank everyone else for joining us today. We're really excited to be sharing the technology that we have and for taking the time to join us for this presentation. So really what we're going to be talking about today is Isoplexis' powerful single cell biology and how we're really helping researchers accelerate the future of this immune medicine. First off, I want to start by highlighting this picture here. You all do have access, or there is an instrument on site there in Dr. Deepak McGrath's lab. It's the larger of the instruments there. We call that the Isolite. And we'll touch on a lot of the capabilities during this presentation. We'll compare us to some of the other technologies, kind of how we fit into the research flow, the different applications that we have, the workflow itself, and then the fully automated system. And then Noah has a great demonstration for the software tool itself as well. So first off, I do want to start off by highlighting a couple quotations out of some recent publications in 2021 that really highlight these two different metrics that we use for our single cell application. So with our unique biology, it's really becoming the standard and accelerating these cell therapies. And with these two quotes, highlights two different novel metrics that we have, or one of them, and rather you've probably heard of the other one, being polyfunctionality. And those are the cells that are secreting multiple cytokines. And then taking it a step further with this novel metric we have is polyfunctional strength index, or PSI as we like to call it, which is really the frequency of those polyfunctional cells and their secretions. And Noah will talk a little bit further about that in the demo presentation, about what that actually means and how you can stratify differences in patients and things of that nature. So in this first publication, Nature Medicine in July of last year, it was actually applied to their CAR products. They said that their research suggests that engineering iteration should be guided by careful studies of single cell CAR polyfunctionality, incorporating cytokine production as a critical quality attribute into their work. So really exciting to see how they applied it there and utilized that polyfunctionality to look at a quality attribute of their CAR products. In the second Nature Medicine publication, it was looking at a TIL therapy, and they said that they assessed the polyfunctional strength index, which is an indicator of the number of cells capable of secreting multiple types of cytokines. PSI reflects the ability of this T cell to carry out multiple functions. It's recognized a metric for potency of cell therapy and efficacy for vaccines. So two different ways that they looked at the data, and just to highlight the two different terms that we'll be touching on quite a bit here. So really what we have here is this suite of products that we're fitting into, and our unique biology and system is really help accelerating these curative cell therapies. In fact, we've really broken into the scene strong, including the one that you all have there. There's over 160 isolates spread across the world now. We're actually over 60% of the comprehensive cancer centers now, and are working with all the big pharma companies across the world, really fitting in all along the R&D pipeline from preclinical to clinical trials. And we're doing this with a lot of great partners across many different research areas. In fact, we have over 55 predictive data sets with many partners across academia, biopharma, therapeutic companies. For example, in COVID-19, we're part of a consortia looking at treatments and vaccines for COVID-19, as that's become kind of a hot point for research areas over the last 2 years. On cancer immunology, which is really our bread and butter, we're working with well-known institutes to characterize responses to new immunotherapies and discover blood-based biomarkers for predicting treatment success. In cellular therapy, or CAR T, we're looking at the potential of a CAR T product success before it's actually infused into the patient. Some of those are just examples of the awesome partners and areas that we've been doing. And we'll take a look at a couple publications specifically across our different product lines and how that's been applied to people's work there. So with that being said, really breaking into the market really quickly and being adopted rather fast. Now over 160 isolates in the field, like I said, so really no longer at this startup stage, and we're really expanding our product line, now have over 300 employees. So it's really a great time to be a part of this presentation, hearing what we're offering, and consider this as a part of incorporating it into your workflow. So I mentioned some of the great partners and institutions that we're actually working with. You can see on the screen some of the great ones that are really using our technology to help accelerate their therapy development. They are across many different areas of that research pipeline, where we produce data in many different cell lines: CAR T, NK cells, macrophages, TCR-T, across allogeneic, autologous, bispecific, and CRISPR indications. So really fitting in a lot of different areas, both within that pipeline as well as models and different cell lines as well. To take a look at really how it is that we are differentiating ourselves from the technology out there, we like to take a look at kind of the status quo technologies and see how we're actually able to take a different approach to identifying some of these secretions within these cells. So the first, kind of way that was looked at is these bulk methods, where really you're taking an average across all the cell types, typically running something from a supernatant where it's collected off cells, something like that. And really, you're just going to be, running that typical assay, taking an average of all of those samples, secretions, and really getting that broad picture that you could be looking for. And what we see is it could be really missing this critical immune cell attribute, or the polyfunctional cells, the small subset of population of cells that are secreting multiple different cytokines. And really what we're looking for is what we've called these superhero or supervillain cells, where we're actually able to take a look a bit further. We kind of make an analogy to it on this slide. If you were to drink some type of smoothie, any type of fruit drink, you'll get this overall flavor, but you don't really know what individually was put in. And that's really what we're taking a look at here, is identifying those single pieces of fruit. You can see it on screen as akin to a blueberry. And you're really able to look at that the secretion phenotype of those cells. We're actually capturing live secretions of these single cells, and then able to take the secretion detail and actually link it to those individual single cells. So we're capturing these superhero cells. And what I mean by superhero cells are really the ones that are orchestrating this immune response. And they orchestrate this immune response through their simultaneous secretions of multiple different cytokines. So I said it was a small population of cells, and typically what we see is they do only count for about 5 to 10% of the cells in this heterogeneous cell population. And these are really the subset of cells that are driving this immune response. And we can capture these individual functions of these single cells through our platform's highly multiplex assay. And we'll talk about that a little bit further, how it works and really what it is. But in this manner, what we're doing is identifying these cells with multiple functions, i.e. the polyfunctional cells. And these are either going to be, the superhero cells, or also the supervillain cells, which are the ones that are suppressing that immune response. So two different ways to look at it: the superheroes that are orchestrating the immune response, or on the other end, the ones that are suppressing that immune response. And so really what we're able to do is identify those cells that produce a potent immune response against a pathological state. We then relay that information into the polyfunctionality, and from there into the polyfunctional strength index metric, which accounts for the frequency of those cells, the levels of their cytokine secretions, and the intensities of those cytokines compared to either a control or a non-responder situation, depending on the case. Now we'll take a look at a couple case studies, see how it's actually been applied in the field and some things, interesting data that they've found. And most of this is utilizing, the PSI or the polyfunctionality of those cells. In this first publication, this is actually referencing that Till's publication in Nature Medicine. This research was actually done at the Duke School of Medicine and the Moffitt Cancer Center, where they were looking at the benefits of using our technology in TILS therapy. In this study, our unique biology is recognized as a standard for product potency and efficacy. This type of predictive metric is really a critical need for the field that we're uniquely providing. And really, we're the only technology currently available that's able to unlock this true functional insight at the single cell level, which is really helping them accelerate their therapy development. So in this study, the group extracted some TILs from lung cancer patients. They then expanded and cultured them ex vivo. They then infused the TILs back into the patients. And then you can see the PSI values of their peripheral CD8 T cells pre and post infusion here. So you can see the differences in the PSI pre and post infusion. And this is really where they said that it was recognized as a metric for potency of the cell therapy and the efficacy of the vaccines. In the second publication, they were using a different one of our applications. So on the first one, it was our secretome assay, where we're actually capturing the live secretions of cells. And this one, this is our intracellular signaling omics panel, where we're actually looking within the cell and capturing the phosphoproteome. I'll highlight a little bit more about what those specific applications are further, but just to share a little insight into the differences and how they're applying it here. So with this one, they were using our intracellular proteome tech to detect changes in signaling in these supervillain tumor cells after treatment in a patient-derived in vivo glioblastoma model, which was known to be resistant to mTOR kinase inhibitors. This single-cell intracellular application was able to detect changes in signaling very early on, anticipating drug resistance long before it was clinically manifested. From this information, what they did was they took some combination therapies, or they identified some combination therapies that resulted in complete and sustained tumor suppression in vivo based on the predictive data that we actually unlocked from that intracellular application and the data that was produced there. Third application is a publication in 2020 in Cell, where these researchers were using our assay on COVID-19 samples. And they uncovered that in moderate to severe cases of COVID-19, they saw an upregulation of inflammatory cytokines in monocytes, which were acting as a supervillain in this case, while simultaneously they saw downregulation of secretion in CD4 helper T cells. The study then went on to further investigate an IL-6 inhibitor. Post-treatment of that IL-6 inhibitor revealed downregulation on the monocytes, upregulation on the CD4 T cells, and ultimately correlated to patients recovering from these severe and moderate cases of COVID-19. One more case study to take a look at was in 2020 in Nature Communications. The Isoplexis technology actually was used and identified this blood-based biomarker, which predicted persistent response and progression-free survival at week 1, well before the radiographic evidence, which was over 8 weeks later. So just an example of really how you can get this predictive data, 8 weeks sooner to allow you to make decisions, potentially answer new questions, or take yourself in a more direct and understood route, rather than having to wait several weeks to get that data. This polyfunctionality really pinpointed the clear choice in combination preclinical therapy, which correlates to persistence in longer-term models. And Tony Rebus, who did this research, was actually quoted in saying that the Isoplexis assay is the most conclusive data that we have obtained in this model system. So great kind of backing from the researcher who was taking up the project here. Just the last publication that we'll take a look at here. And now we'll actually look at the workflow so that you can get an idea of exactly how it is that the cells are produced or put onto this application. So to start it off, we are an ELISA-based application. We do a sandwich ELISA at the core of our technology. What's unique about the application, aside from the single cell nature of the application, is that once you load your cells or your sample onto the chip, everything is fully automated in the ELISA workflow. So we have microfluidics on board the instrument. You load your single cells or your samples onto the chip, and then you press go, and that instrument takes care of the rest of the ELISA for you. To take a look at the workflow itself, you can see a couple boxes at the different steps. For the sample preparation, you're going to start with, for the example on screen here, you can start with your cryopreserved samples. You'll then recover them overnight to really, allow those cells to wake up. Then you're going to enrich for a sample cell population. So for example, if you were looking at CD8 T cells and the response that you would get from those T cells, you would use an enrichment kit to get down to the CD8 T cells. For an example, we typically have our protocols using the Miltenyi magnetic bead system. So you would use that for a positive selection, positive enrichment of those CD8 T cells. You would then do some type of stimulation. We have many different protocols. You can do a biologically relevant stimulation, or you can go the route of PMA inomycin. Many different applications or routes you can take there, but you do stimulate to wake those cells up, get them secreting so that we can capture those on board the chip. You then stain those cells before you load them onto the chip so that our instrument can actually identify where on the chip and in those wells the single cells are located. Then you load the sample onto the chip, you put the chip onto the instrument, and then the fully automated assay runs the system at biological conditions. For the secretome assay or for the intercellular assay, quickly starts capturing the phosphoproteins on board that chip. The results are then analyzed on board the built-in software system, which we call Isospec. Noah will be doing an excellent presentation of really what you can get out of that software system. Really intuitive and a great presentation that will be following my talk here. Now we'll take a look at how the chips themselves actually work. So really what we do and how we're able to unlock this powerful biology is through what we call this proteomic barcoding. And how that works is you load your sample onto the chip. You can load your single cells for the single cell application, or we also have a bulk application that we added last year where you're going to be loading, supernatant, some type of cell-free solution. But either way, you load your sample onto the chip, and then each sample is captured in one of the respective chambers, whether that be micro chambers for the single cell application or macro chambers for the bulk application. From there, either the enriched single cells or the supernatant on the bulk chip flow across the chip, and then each chamber has access to this full multiplex barcoding where they're able to run this sandwich ELISA in parallel for each sample and each chamber in a fully automated manner. What we're able to do is up to a 32-plex multiplex cytokine analysis, depending on the panel that you choose. And to that point, we currently have 13 different panels available. 14 now. We released a new NK cell panel last week, which we're really excited about. And then currently have 19 other panels currently in development. So really trying to expand this product pipeline and the access or the researchers that will fit into. We're trying to expand that rapidly and really support all the customers that are out there in the field. Kind of wrapping up the workflow at the back end of it, we have the Isospec software, which is our onboard data analysis tool. It really provides you publication-ready insights on the same day as that run is finished. A lot of the graphs and things that were shared in the publications a few slides ago were actually generated in this software tool. It's very intuitive, and really we do a lot of the work for you. All you really have to do is annotate the samples, but on the back end, the data is auto-gated, and the process is really happening as the machine is finishing up its run. It pushes out the data. Once you annotate, you press start, and then it quickly will run through the data and have it ready to be parsed through, where you can look at these publication-ready figures using this push button interface, and it allows you to evaluate all the levels of the analytes from your run. You can see some of that on the left there. You can also use a lot of our different biomarker visualizations, which can be easily generated simply by pushing the respective buttons at the top row of the screen. This really provides you the ability to stratify your data, uncover the drivers of response, and deliver these same-day correlative insights. where you're really gaining the ability to access this powerful data right away, rather than really having to consider outsourcing or waiting for someone to run through the data yourself. So you really get, yourself the ability to look at this data, someone who understands it. So adding into that layer of just having access to that data and being able to understand really what you're looking at from day one. Noah will do a great presentation there, and we'll do that quickly following after this. Finally, I wanted to touch on the 3 different families of chips that we have, which you can see here. The high-plex walkaway immunoassay is the one that I referred to as the bulk application. On that application, you're going to be loading supernatant, some type of cell-free solution, serum. We've done tracheal lavage fluid. There really are many different ways you can load onto that chip, but it is just going to be some type of cytokine-containing solution free of cells. You can use this in an application to kind of optimize for single cell, or if you're looking for relative changes in cytokine profiles, that's a great route as well. It's an ultra low volume solution, so you only need 11 microliters of sample to load onto that chip. Taking it a step further and really diving into the single cell level of the application is this functional immune landscaping. That's our secretome assay. So really what we're doing there is you're able to capture the functional phenotype of this heterogeneous single cell population and identify these superhero cells orchestrating that immune response. How we're able to do that is we load live single cells onto the chip. Onboard the instrument, there's an incubation portion that we're actually going to incubate these cells at physiological conditions for about 12 to 13 hours. And along that process, we're capturing the live secretions of those single cells and then mapping those secretions back to which single cells they came from. The last application and our newest application is the intracellular signaling omics. Still the single cell level of application, but instead of capturing the live secretions of these single cells, we're actually going to be using it as like this multimodal pathway of analysis for protein phosphorylation to gain insight to what's going on inside of the cell. So rather than a live cell assay, we're actually flowing a lysis buffer through early on in the assay, breaking open the contents of those cells, and then capturing the phosphoproteins to really get an idea of what's happening within those cells. So really what we have is kind of the Swiss Army knife of proteomics, where you get this fully end-to-end automation application, where you can take a couple different looks, both at the bulk and then single cell level, at what's going on with your cells and really get a better understanding of either the secretions or the phosphoproteins within those cells themselves. To really wrap it up, we're delivering this, impactful data to create more potent, durable therapeutics. We have many other case studies to share. If you want to have a follow-up meeting, we can talk about some that are, maybe more applicable to the cell types or work that you're actually doing. But here's an example of some of the therapeutics or cell types that we're actually doing. And we actually have recently released this functional cell library, which kind of highlights the different areas that researchers have actually applied to this technology, broken down by different cell type, different models, whether that be a mouse, human, or non-human primate. And so if you were interested in taking a look at that, feel free to reach out to me after this presentation and we can talk to it. This is the kind of wrap up portion of my presentation itself. After Noah's completed, we'll talk a little bit more about this exciting grant program that we're going to be hosting, allowing some of you free access to generate some pilot data. We'll talk about that a little bit further, but for now, I will actually hand this off to Noah to begin his portion of the demo presentation. Thanks.
Awesome. Thank you so much, Brad. Can you hear me?
Yes, we can.
Perfect. Hi, everybody. It's great to meet you all. My name is Noah Vanderhyde. I am the FAS for the state of Michigan, as well as Illinois and Indiana. And today I'm going to be going through our demo software utilizing our new, actually, IsoSpeak 2.9 update. Something that is really, really nice about this update is it's consolidating a lot of the older key factors that we have down into 3 tabs instead of 5. So we're really increasing that ease of use on the platform. And we also have some different UI changes. So we have this nicer dark mode, and it's really, really great to parse through. Right as Brad, was saying, you're going to be getting all of the data that's automatically analyzed on the instrument, but then you're going to have to go put it into the software. So after, 12, 24 hours, of your single cell or codeplex data being analyzed and going through the laser detection, you're going to take it and put it onto your Isospec laptop. Here, what we have is our CAR-T demo, and we're looking at the single-cell secretome chip family. So these are the secreted cytokines that you'd be detecting from single cells. And we're looking in our adaptive immune panel on human. We have a bunch of different panels and different species that we have them in. So this is the one that is specified for this demo. In Isospec, we have 3 different areas: your project info, which is your data annotation section, your data processing, which is where that walkaway software analysis really lives, and then the data analysis section. Here we have all of our publication ready figures and tables ready to go to be exported at the highest PNG level. To dive right in, we'll take a look at the annotation section. And here you can see it's pretty easy to parse through. Unlike flow cytometry and some of the other legacy technologies, you don't really have to sit at your computer and go through a lot of gating processes, or you don't have to sit there and do a whole bunch of standard curves. All of the information is ready to go right in Isospec by the time you even put it into the instrument. What you would typically do is you'd press the import button up at the top left, and you would import your raw instrument data, or you can import different .zips that you already had generated data from to create a larger data set. The data set that we're going to be looking at today has 24 chips in total. So this can be 6 4-chip runs, or 3 8-chip runs all in a row. And this data set is actually modeled off of one of our publications, where we have 6 different donors in the CAR-T realm. Donors 1 through 3 were non-responders to CAR-T treatment, and then donors 4 through 6 are responders to CAR-T treatment. Additionally, this group who was running this small clinical study wanted to see in the manufacturing realm if the control method or the old method of stimulation and manufacturing was better for CAR-Ts, or using this new target stimulated stimulation assay. Moving left to right, we over here had the chip IDs. Each chip will have its own numerical ID printed right onto it that's scanned by the instrument. If you or one of your lab members or several of your lab members have been working on the assays at a time, you can type in who specifically did the assay. So I can type in my initials here, NSV, and I can type in Brad's BA. So you can really get to see who was doing those experiments. From there, if there's any notes such as the viability dropping or a couple of key things about the biology that you need to put in, we have a notes text field here that's easy to use. Next up, this is a laser-based detection platform, and so we are going to be using either general membrane stains or a few marker-specific stains to detect our cells of interest. In the workflow, upstream, you'll be either magnetically enriching your cells or using some FACS sorting to pull your cells types of interest down from your PBMC populations or your TIL populations. Here, we're currently looking at only CD4 and CD8 T cells. But if we type into the cell types here, we can type in C. We have CARs, CD14, CD154, CD3, and so on. As we're starting to build this functional cell library, we really want to encourage a lot of users to bring in their own types of cells of interest into the software. Here in just the secretion realm, we have only our targets, endothelial cells, and fibroblasts and more. But when we go into the intracellular tumor proteome version of the Isospec software, you can pull down if it's your glioblastoma, your sarcoma, and more. Over here, we have our annotation fields for donors, donor groups, and stimulants. And then over here on the far right, we have areas that you can normalize to your viability or your transduction efficiency if you're working in CARs or TCR or any engineered products. Once you've annotated pretty much everything here, you're going to head over to the data processing tab. And really, the data analysis in Isospec is so simple. You'll just select every experiment you want to analyze. You'll press the Analyze button, save your projects and experiments, and walk away. In this case, there's no data to be analyzed since it's a demo data set, but I just want you to imagine that this progress bar is empty right now and all of these fields are empty. Starting at 10%, the software will go through and start detecting these cells that are in these single microchambers all alone, and gating out any microchambers that are containing 2 or more cells, and then taking into the background. And that background is generated from microchambers containing 0 cells. As it moves from 10 to 100% and going through the signal imaging steps and the chip alignment steps for the software, it's going to tell you that if it's completed, and it's going to spit out a couple quality score metrics for you. We also have this warning section here that has to do with any laser differences or any issues that the software technically finds. Over here with the 2 quality score metrics, we have our quality cells, which are the number of single cells that were generated in this data set per chip. And then we have the ISO-Q score. Since IsoSpeak and its analysis process is all automated, we have an automated quality assurance metric that's also with the analysis. This takes into account a lot of different things about the chip, such as your sensitivity, any debris that was detected on the chip, and even down to just a signal saturation. Since all of our ISO-Q scores are high, this means the data is of extremely high quality, and we can move on to the data analysis section. When we get over to here, we have a couple different things that you can sort through. Really at Isoplexis, we believe in democratizing your data sets so we can turn on and off exactly what we want to see or what we don't want to see. So we can toggle off, any only secreted cytokines in the data set. Or, when we're doing our analysis, hey, I only want to look at the CD8s. And as you can see, IsoSpeak will automatically update some of these text fields for you. So you're looking at the most accurate representation of your data. Before we really dive into the rest of the figures and everything, I just want to show you how many data points that are generated inside of these data sets. From 24 chips, this number right here is the number of cells generated. So we have 14,000 cells from this data set multiplied by our panel of 32 cytokines specifically on this panel, and then 2 extra metrics, the polyfunctionality and PSI. That's over 14,000 times 34 data points from just these analyses. There's a lot of data to parse through, but this data is extremely powerful and is really characterizing of your polyfunctional subsets of cells. In this case, for the software demo, what we're going to do is we're only going to be looking at our responders, so donors 4, 5, and 6. We're also only going to be looking at only CD8 T cells, and we're only going to be looking at the secreted cytokines here. So, from 14,000 plus cells, we get down to about 3,800 that we're really going to parse through in this data set. With all of this updating, you can see that we have all of these different cytokines that were observed across the data set. In previous versions of IsoSpeak, you had to go through the table section as well as the single chart data, which is the per sample information, and then the aggregate data all in 3 separate tabs. But here we have them all in one window. So here, if you want to see, your target information based on the samples or the chips, you can have them here. Or if you want to see them all in a representative bar graph of one another, they'll be right next to it, or on a table. And this table can also be exported as a .csv to take into other, statistical programs such as R or SPSS or more. When we're looking at these data sets, we can see that in the secretion frequency tab, the cytokines that are detected in the target stimulated conditions are much more increased compared to the control conditions. From the user, this is something that they really wanted to see and gave them that confidence in moving forward with this target stimulation method for their manufacturing of their cars. We're also going to take it only down to donors 4 just for now, so we can see some of these key differences. In donor 4's control, you can see that there's a lot of granzyme B detected and a little bit of IL-8, but even in the target stimulated, there's such an upregulation of cytokines that's detected. Down here in the key, we break our cytokines up into 5 dominant functional groups: effector, chemoattractive, stimulatory, regulatory, and we're missing just one right now, and that's inflammatory. We have a few different panels that will characterize data differently inside of IsoSpeak. In the adaptive immune panel, it'll group them by these dominant functional groups, but in the inflammatory panel, it'll group them by Th1, Th2, or Th17 pro-inflammatory pathways. Moving on to the signal intensity chart, we have them in representative of a bar graph, but we also have them in signal distribution. This is typically the better way of taking a look at your signal that's generated from the chips. And when you zoom in here, you can see that there's these dots. Each one of these dots is a single cell that is contributed to the fluorescence of that cytokine. This bar right here is our bar of the background threshold that we need to be surpassing to, call it a significantly secreted cytokine. And then we have an average here, which is 3 standard deviations above that background threshold. What we can see here too on the top is the secretion frequency percentages on top instead of just here on this bar chart. And here we can see that there's a lot of signal being generated from the target stimulated. And we can even take a look at it in a representative scatter plot if we bring in donors 5 and 6 as well. Moving into polyfunctionality, which is a key metric here at Isoplexis, these are your cells that are secreting more than 2 cytokines at any given point. These cells are really what's driving your polyfunctional response in what could be, cancer tumor burden clearance or clinical efficacy of your engineered product in vivo. This is something that's really, really great and is an automated metric that you get when utilizing this product. What you see here is that there's an upregulation in polyfunctionality between your controls and your target stimulated conditions, meaning in this case that there's a lot more activity when these are manufactured with the actual target. This is something that the client really liked, and what you can also do is you can play around with how this data is shown. So currently we're only breaking it up by control versus target stimulated, but if we want to, we can sort it by the donor group, or we can sort it by the donor. So now we can have each donor right next to one another. You can also change the max graph value for row. So if you see it's just above 13, we can type in 14 and it will automatically change for you. You can also change the labels of these samples, which will affect how they're labeled down here. We also have them information stored in a pie chart. And this is the more sample-based information section, as well as the table for the polyfunctionality. As Brad was mentioning earlier, in a lot of publications, you might see this PSI metric, and this polyfunctional strength index score is going to tell you really what cytokines and cells are driving this polyfunctional response in a variety of different ways. PSI is a relative metric and it can be used to apply to many different modalities of research. What you typically see here as well is that there's an upregulation in PSI along with polyfunctionality between control and target stimulated conditions. If we break it up here in the bar graph grid, what we can also see is the contribution to the overall PSI. So not only do we have the individual scores as an aggregate here, we can say which ones are really taking into that account of polyfunctionality. So in this case of donor 4's, IL-5 is contributing the most to target stimulation. In donor 6, we have more of MIP1 alpha that's contributing here, and so on. In the polyfunctional groups, you may have seen this heat map before. This is the type of cytokines and the total breakdown of the profiles of their cells. If we scale it back to just donor 4, we can see that there's a lot of polyfunctional responses that are happening in these cells, which is really, really great. And these are also unique subsets of cells that are secreting, in this case, 8 cytokines at a time, 7 cytokines at a time, and more. This is a heat map, so it's a really great way to visualize this data. But if you really want to get into the granular specifics of the percentage of those subpopulations, we can then go to the bar graph and see those percentages here. And these are a really great way to break down the data because you can see the exact percentage, whereas here it's just a percentage overall. A really other great way to taking a look at your data in a 2D way is utilizing the PAT-PCA. And this PAT-PCA is going to correlate the size to the percentage of that functional group. And then the overall color weight is the type or the donor or the group of cells that are contributing solely to that subset. So if we zoom in here, we can see that these subsets of cells are relatively small, but they're very polyfunctional in nature. So this one's secreting 5 cytokines at a time and mainly comprised of this green, which is donor 5's target stimulated. And orange is donor 4's target stimulated, so we can see exactly what they're contributing to here. All of this data can also be taken into account in the table. We also have a lot of 3D ways to visualize your data. This one's a 2D t-SNE that's generated, but we also have a 3D t-SNE where we can take a look in a 3D manner, really what the data is telling us. What we can see here is that these are the controls from, CD8 T cells from donor 6. But if we move to this group, we can see that these are the target stimulated CD8s from donor 6. We also have the information with donor 4, donor 5, and donor 6 included in this data set. But you can also choose how to compare these data points by. So if you want to compare them by their dominant functional group, you would just have to change the compare data points section here. Along with this 3D space, you can really turn around and view it in a different way so you can try to find the best groupings possible. And over here on the top right of this section, we have our dominant functional groups represented. In the UMAP, this is more of a global way to sort these cytokines together in these cells, but you can do the same exact thing as your 2D t-SNE here. And then over here in the final section, we have the replicate correlations. Unfortunately, there's not any replicates inside this data set, but if there are, we can see that Pearson's R score that's associated with them. there's a lot of things that you can do inside of Isospec, and coming from a flow background, this was a really great way for us to process this data and then get the key insights out. It's really not about taking the best screenshot anymore, or really messing with the data to get like the high PNG format. You just press the export button at the top right, and you can export your tables, your graphs, your keys all in one section. So if I want to just select only tables, I can select the tables option. Or if I just want to see the graphs, I can just export the graphs. Really, with that is the Isospec software in general. I'm more than happy to take any questions about Isospec or the actual experiment workflow that you would be utilizing before going to Isospec, or generally any other questions about the instrument. Thank you so much, and I can toss it back over to Brad.
Thanks, Noah. We got a couple questions in the chat for you as well.
Perfect. Would you prefer to go through kind of the grant challenge, or would you like me to answer the questions here?
So let's answer the questions there, and then we'll talk about the grant challenge. And hopefully the answering the questions will give an idea of exactly what they can do with the grant challenge itself.
Perfect. Yeah, so I have a question from Dana King. Is there any normalization or thresholding prior to calculating polyfunctionality from the raw signal intensities? And the answer is yes. So essentially what we're doing with the signal intensities is observing to see if it's above, that threshold for that given cytokine on a per cell basis. So if a cell, secretes just under the minimum cytokine level for the RFU to be below the background, it will not count as the secreted cytokine for that cell. And the polyfunctional strength index score is coming from the signal intensities of those cells multiplied by the percentage of polyfunctionality per sample. So it's a really great question. To give you the short answer right now, yes, there is. From Amanda, how many cells can be loaded into the functional immune landscaping type chip? That answer is you're going to be loading 30,000 cells per chip. And from those 30,000 cells, you're going to be in the ballpark of getting, between 200 cells to all the way up to 1,500 cells per chip. You do have to load them as single cells in suspension in RPMI. So in some cases, the single cell chips for adaptive immune for T cells, you would load in at 30,000 cells and 30 microliters, but for your monocytes and macrophages and other granulocytes, you would load them in at 40 microliters at a concentration of 750,000 cells per mil. We load a homogeneous mix per chip. So if you were going through a experiment where you're selecting for your CD4 and CD8 subsets, each chip would have its own subpopulation. So on one chip, you would have your CD4 stimulated cells. On a second chip, you would have your CD4 unstimulated or control. And then the other two would be the CD8s and their own chips each. You can do a variety of different cells. We're building this functional immune cell library. So we have a lot of protocols for T cells, monocytes, macrophages can also be utilized from there. We have a new NK natural killer cell panel that has actually just been recently released. Tumor associated fibroblasts and endothelial cells. And then we have all of those same cell types, including engineered cell types for human, mouse, and non-human primate currently. You can load a heterogeneous mix onto the population, but that would require some dual staining efforts. So you might have to do a surface stain for one of the markers on your cells, and then going to do a cell membrane stain from there. And then Dana also asked, are there any replicates recommended for these assays? Yes, we would definitely recommend running replicates per assay. That does not necessarily mean that you have to have replicates to publish. We've had a lot of groups, just do a 4-chip run and get some really great insights from that, and then go ahead to publish in, high impact journals like JCI. Typically, the replicate or the variability that you would see between them is about in the ballpark of 18 to 20-ish% in inter-assay and intra-assay runs. So if you're in between instrument runs or between instrument chips. And that's why we recommend running replicates to really break down that variance together. And Amanda is also asking, so if you are loading stimulated and unstimulated T cells, you would load with the stimulated media. In some cases, you would. That would be how we used to have our NK cell protocol. But what you would do in the stimulation process is you would stimulate your cells in vitro on like a 96-well plate. From there, you would then take up the supernatants, including your cells, spin it out, and then you would aspirate that supernatant that contains that stimulation, as well as the cytokines that were secreted during that stimulation assay. From there, you would just plop them into an Eppendorf tube and you can freeze them down at -80 to run some population assays on them. From there, we would rinse our cells to get any of that serum off prior to going to the staining sections. And it's also dependent on the stimulation times as well. We have a couple different protocols for T cells where we have an 1.5 hour activation cocktail stimulation from BioLegend. And we also have a 24-hour CD3 plate bound and CD28 in suspension stimulation for 24 hours. Dependent on, really what your interest is with your stimulation method or media, you can always contact me. A recent question I was asked is, could I use Transact compared to a plate bound CD3 versus CD28 stimulation? Really, as your field application scientist, I'm here to help you answer all the questions and help you design that experiment so we can give you the greatest chance of, getting some really great data from there. In terms of protocols, we have a variety of different protocols. If you go to the website, there will be under the, I believe, the resources tab, and then you will just go to the instrument. I think it's instrumentation, and then from there we can have them. I can also pop a link right in the chat while Brad talks about some of this stuff.
Thank you.
Yeah, absolutely.
Awesome. Thanks, Noah. And then Amanda, I can answer the question of will the core of this machine or will we only need to pay per run and ship? This is something that we will need to have the machine purchased into our lab. So great question. Currently, the IsoLite, which is our high throughput kind of core application or core instrument, is currently located in Dr. Deepak McGrath's lab and the north part of campus in the engineering side of things. Forget what the actual name of the building is, but I have been working with Dr. DeGrasse the last couple of weeks to figure out pricing to use it as a shared resource. So people will have access to it. It is in a private lab though. So if there are, hopefully a lot of you interested in this, it may get to the point of saturation on use. Obviously something to explore further down the road if it does get there, but we do have a smaller instrument, the IsoSpark. It is more of a personalized lab instrument, which Cole Sweeney is the instrument rep for Isoplexis for University of Michigan. He can have that conversation if you all are interested in going that route. But for now, there is a IsoLite that is going to act as a shared application for people that are interested, which takes me to the point of the exciting grant application that we're going to be offering, which we're working, we're coordinating with Dr. DeGrasse there. So what we're going to be offering is, I will drop the link into the chat box now. And what we're going to be offering is basically the ability for 3 winners to get some pilot data and run up to 8 chips of your choice on any samples. And what we'll be doing is taking basically grant applications from whoever wants to submit from that landing page that I just posted. A couple things to fill out there. From there, where they will be screened on our behalf, and then we're going to send them to the University of Michigan and the single cell program to ultimately select the final 3 winners. What you'll get from winning is basically you'll get the FAS support from Noah and then myself. I will work with you to figure out, the best way to apply these 8 chips to your work and get you some initial pilot data. We'll support you through the whole process, get you trained on the instrument, show you how to parse through the data, get you comfortable with the workflow and everything, and be there to support you on site for all of those portions of the work. Additionally, we will also, meet to make sure that the project plan is in order and everything like that. So we're going to provide great support throughout the entirety of the process, as well as the back end with generating and parsing through the data. And you'll be able to win up to 8 chips per winner to run whatever cell type you want. So as Noah mentioned, we have human, mouse, non-human primate, and then many different cell lines. We have an adaptive panel, innate panel, natural killer cell panel. We have an intracellular tumor signaling panel. So many different applications that we can use and explore there. And we're really excited to be offering that to the university. Applications are now open to be submitted from that to that link, and we'll be collecting them through the end of this month. And then we'll be announcing it, I think, first week or second week of next month. And then we'll begin the process of really working with you all and the winners to work through that. Additionally, if you do apply and unfortunately are not selected, I'll be offering kind of an exclusive discounting structure if you're interested in going that route as well. One last thing that I will mention is myself and Cole Sweeney will actually be on site Thursday and Friday of this week. If you all would like to have a further conversation either about the grant challenge or just the technology in general, or just want to talk and we can learn a little bit about your work, we always love those conversations as well. So thank you again to everyone for joining us. Looks like we do have one more question. What if you need to run some samples now? Yeah, great question. So what you can do now is we can set up a private meeting following this. I'll actually post my email in here so that you can reach out to me. And then what we can do is quickly work through kind of figuring out the questions that you want to answer, figuring out which chips, experimental design through the support of Noah and myself. We can quickly get you kind of a plan to get chips shipped out to you and figure out how the purchasing and everything like that will go. Loop in Dr. McGrath, and we quickly are able to provide chips in about a 2-week lead time. So if you were wanting to run, or had an interesting question you wanted to explore and had some type of timeline, we would be able to begin working with you probably within 2 to 3 weeks. So we do have a great support system, very local to you all, and quick turn times on our chip products. So yeah, great question there, and we can begin planning those projects quickly if there is a timeline issue. But thanks for that question. I will go ahead and wrap things up here unless anyone else has any further questions. Thank you, Justin. Thank you, Vanessa. Thank you, Dr. Keller. Great to have all of you here. It was a well-attended event, so I hope you all were able to take some great information away from this. If you all do want to have follow-up conversations, please do not hesitate to reach out to me. Like I said, I will be on site all day Thursday and all day Friday taking meetings, and we'll just be walking around campus as well. So thank you again. It was great to give this presentation to you all, and I hope you have a great rest of your day.
Great. Thank you so much, Brad, Noah. Looking forward to seeing some cool grant applications come through to use these products.
Absolutely.
Thanks, everybody. We hope you have a nice afternoon.